Hello, grade 9 students. In this presentation, we're going to talk about the basic mechanism of sexual reproduction, about mitosis and meiosis. Let's start with the small information about biology. Biology is the only science where multiplication and division they have the same meaning. We have the increased number of cells. Mitosis. Mitosis is the process by which the somatic cells, pay attention to the word somatic, meaning they deploy to and they undergo the process to produce new daughter cells. These daughter cells, they are identical, they are similar to the mother cell, having the same genetic information from one generation to another. Why do the body cells need to reproduce? First, they need to increase the cellular number and allow the growth of individual, like the case of the zygote to be an embryo, or to renew the body's tissues and replace the old or the injured or the dead cells. Mitosis is found in the body but is not always occurring. This is the mitosis. It may be divided into interphase that start with G1 as G2 and mitosis, prophase, metaphase, anaphase, and telophase. This is the cycle of the interphase G1 as G2. S is called the synthesis, where DNA synthesis and replication took place. We have mitosis and cytokinesis, where the cell is going to divide. So as I said, we have G1, first gap, cell is doing its everyday job. Then we have S phase, synthesis of DNA, where DNA is replicated. G2, preparation for mitosis. Mitosis, we have prophase, metaphase, anaphase, and telophase to produce two daughter cells that are identical to the mother cell. Interphase, we have chromatin like the hair, like structure as I said in class. You have centrosomes, nucleosis, nuclear membrane, plasma membrane. We have the chromatin, and this is called the interphase. Prophase. Prophase, the chromatin condense into chromosomes. These chromosomes, they are of two chromatids each, and we have centrosomes change into asters. The nuclear membrane disintegrates. Mitotic spindle fibers also they are formed. Centrosomes change into asters, as I said, and move to opposite pole of the cell. Chromatin condense so that each chromosome is made up of two chromatids each. Second phase, it's metaphase. Metaphase, chromosome, they line in the middle of the cell, forming what's known by equatorial plate. Anaphase. Anaphase, sister chromatids move to the opposite pole of the cell, moving what is known by polar ascension. This, at the end of this stage, we have two groups of chromosomes, each made of one chromatid each at the opposite pole of the cell. Last phase of the mitosis is telophase and cytokinesis. Telophase, their chromosome, they form two identical groups at the opposite pole of the cell. This chromosome will decondense back into the chromatin form. Nuclear membrane reform around each set of chromosomes. The spindle fibers disintegrate and the asters return to the centrosome state. Cell membrane constrict, forming what's known by cleavage furrow that divide the mother cells into two daughter cells. You have to know that the process when the cytoplasmic membrane starts to constrict, cleaving the cell into two daughter cells, is called as cytokinesis. At any stage during mitosis, the cell has two chromosomes. The two daughter cells obtained at every cell cycle, they are identical, they are the same, having the same genetic information as the mother cell. They are diploid, having the same genetic information. Note that in plant cells, there is no asters, no constriction due to rigid cellulose wall, so a new cellulose wall will be formed at the middle of the cell, leading to two daughter cells. This is very important. There is no asters in the plant cell. Moving now to meiosis. Meiosis is the process by which the mother cell of gamete 2N produces the gametes haploid 1N. Before meiosis begins, the mother cell goes through an interphase in preparation for meiosis that includes two successive divisions. We have meiosis 1 and we have meiosis 2. Meiosis 1 is called reductional division and meiosis 2 is called equational. Meiosis 1, prophase 1. The nuclear membrane disintegrates. The spindle fibers start to form. The centrosomes change into asters. Chromatin condense so that the individual chromosome they are made of two chromatids each. Here we have the difference. Homologous chromosome they pair up, synapse. We have it's known by tetrads. This increases the chance of crossing over between the necessary chromatids of the paired homologous chromosome. This is metaphase, and this is what's known by the tetrads. Tetrads they line up at the equatorial plate during metaphase 1 of the spindle at the equator of the cell. Meiosis 1, anaphase 1. Spindle fibers they pull the homologous chromosomes of each tetrad apart. Each chromosome moves to opposite pole of the cell, making what is known by polar ascension. Late anaphase, the result will be two groups of chromosomes, each made up of two chromatids at the opposite poles of the cell. Meiosis 1, telophase 1. Telophase 1, cell membrane constrict, forming the cleavage furrow, so two daughter cells are obtained, where each carry one copy of each chromosome, made up of two chromatids each. Obtained daughter cells, they are haploid, number of chromosomes is reduced, that's why it's called reductional division. Daughter cells, they are different from each other, and they are different from the mother cells. So chromosomes form two groups, the spindle fiber disintegrates, nuclear membrane reform, and no interface you have to know between meiosis 1 and meiosis 2. Now let's move to meiosis 2. Meiosis 2 we have two daughter cells. So mitotic spindle fiber also start to reform. Centrosome change into asters. Nuclear membrane disintegrates. Metaphase 2 of meiosis 2, here chromosomes line up, and you have to know that the meiosis 2 is very similar in its events to the mitosis process. Anaphase 2, the sister chromatids they are pulled apart, each chromatid moves to the opposite pole of the cell, forming what's known by polar ascension. And late anaphase, the result will be two groups of chromosomes, each made up of one chromatid at the opposite poles of the cells. Telophase 2 and cytokinesis. The chromosomes form two groups at the opposite poles of the cell. These chromosomes will decondense back into the chromatin form. The nuclear membranes start to form around each set of chromosomes. The spindle fibers disintegrates and the asters return to the centrosome state. And this is the processes that occur during mitosis and meiosis, and you have to review them very well.